תינתן על ידי דוקטור אלכס שיין, מנהל פיתוח RNA בחברת ביוסייאנב, שהיא חברה צעירה שהוקמה על ידי שני הורים לילדים חולי דושן, שהינם גם חברי עמותת עדי וממקימיה, אורן עוזרי ואלון חזן, הנמצאים כאן איתנו היום. החברה הוקמה במטרה למצוא תרופה גנטית למחלת בניהם ולכל שאר ילדי הדושן. דוקטור שיין, בוגר תואר שלישי בביולוגיה של RNA בטכניון בחיפה, עשה את הפוסט-דוקטורט שלו בקופנהגן ועבד במכון מחקר למדעי החיים ביוקוהמה. בחברת דוקטור שיין אחראי על הפיכת החומר המעבדתי לתרופה. פליז, דוקטור שיין. אני רוצה להעביר לך השקף שלך, אבל אני...
את הולכת קדימה.
זה לא שלך, נכון?
זה שלי.
זה שלך.
אבל זה הסוף.
אוקיי, אז תגיד לי מתי לעצור.
Right, now that we have the right slides, we can start. Thanks to the organizers and thanks for everyone for staying this long, even though it's almost a lunch break. And I'm going to present a very different approach to treatment of genetic disease, which is messenger RNA therapy. And Art Bioscience was really found almost 3 years ago, and we work in Rehovot. It was founded by two people who are sitting here and who have kids suffering from DMD. We're collaborating a lot with different partners in industry and in academia. And then DMD was actually introduced before, and I believe that there is no need to stop here, as we all unfortunately know what is dystrophin and why is it important. But we've had a really good presentation about gene therapy. So there is gene therapy that is being developed, there is stem cell therapy, which is trying to introduce healthy cells into the sick muscle. And then basically, there is another therapy which tries to bypass the affected site on the DNA. And that's it. And here comes messenger RNA therapy. But before going to that, I might say a few words of what is messenger RNA, even though it was also introduced by Yoram before. So when we think about mRNA, or if we look for what is mRNA, we usually see something like that, which is a figure showing where the mRNA comes. But that might not be very informative, at least, for those who don't work with mRNA all the time. So I made this figure to introduce the mRNA in a better way. So there was also a talk about the book. So DNA is a book which stores all the information about what we're supposed to look like. But that book is made of stone. It's very heavy and it cannot be moved. It stays in the nucleus. So in order to get any information about that book, one must transfer that information into that factory, which makes protein in the cytoplasm. In order to do this, part of that book must be rewritten into a message by the same chemical letters as the DNA is made of. So this message is light and it's much more compact, but it's fragile as well. But nevertheless, it can be transferred to the cytoplasm and then that factory may start working and producing the product, which is protein in this case. So mRNA is a message. And coming back to DMD, those red picks at the DNA are exons, and somewhere in there may be mutation. We've heard about different mutations, which is a problem. So it is difficult to find a universal treatment for all mutations that occur in the gene. So what happens is that the mRNA is broken. So it's produced, but it's not functional. It cannot be used to make dystrophin. And then the protein is missing. What we do is we make that mRNA, the message, in the lab, which is here on the right. And then we pack it into a nanoparticle, into, well, a vesicle in a way, which protects the mRNA and also helps it to get through the cell membrane. So when it gets there, because it's a healthy mRNA can participate in translation, it brings the right message into the cell, and then it can drive the translation and restore the protein. Apparently, this approach does not require any changes. It does not depend on mutation, which each individual patient has. We can treat anyone with that. It works in stationary cells, which is important because, I think I was asked a number of times, why don't we use the DNA for that matter? And DNA has to get into the nucleus in order to function, which is not the case with mRNA that works in the cytoplasm. And yes, there is no genome editing that is introduced. So here is what mRNA therapy looks like. And this slide here shows that it also has disadvantages or difficulties. So we are not restricted, for example, for the mRNA size, or let's say we are less restricted than in the case of gene therapy. But it's still difficult to make very long mRNA, such as the one of dystrophin. Those green cycles actually show that we are able to overcome those obstacles and move to production of mRNA and testing it in vivo models and in clinic afterwards. Just a word about the mRNA itself, because I'm going to come to that later. This is what mRNA looks like. So in the middle, there is a coding sequence, the actual message, which tells what the protein would be like. But it is also surrounded by the regions that are not encoding for anything, but they are still very important for the stability of mRNA and for translation, because the protein-producing machinery has to recognize those sequences and react to them appropriately. It also has this A250, which is a tail with adenosines that is important for functioning of mRNA. And it has the cap on the other side that is very important for stability. So in order to make a proper functional mRNA, we have to bring all those elements together, which is what we do by the help of the technologies that we are able to use. So up there is a scheme of human dystrophin, the protein, the full-length protein. The RNA that we make encodes for dystrophin that is shown below. As you can see, it's almost full-length protein. It contains all functional domains and is also based on clinics because a very similar form exists in a number of patients with a very mild form of the disease, or even with those without symptoms at all. Those figures show what the mRNA looks like when we, well, check is for the integrity. So those bands are molecules of RNA that run on a gel. We can see them, we can control their size. Then we test how the mRNA works in making the protein, initially in a cell-free system. So we introduce the mRNA into the isolated environment that can make proteins, and that's what we see. Those bands now show protein. The last band on the right is a full-length dystrophin of the mouse. So our protein is a bit lower because it's smaller. So we have the right mRNA and it's functional. When we transfect the cells with that mRNA, we can detect it staying in the cell for 96 hours, maybe longer, but we didn't test that. We can also see the protein that is being expressed. Dystrophin that is encoded by our mRNA is also staying in the cell for about 96 hours. 101 is the one which I've shown before, encoding for the longest form of dystrophin. There is also 102, which encodes for a shorter form. So that encodes for a smaller protein according to the markers. But it's still there in the cell for up to 96 hours. The next step is in vivo test. And here we inject the mRNA into the muscle of MDX mice, which do not produce their own dystrophin. So after the injection, we can detect the RNA in the muscle. This is 24 hours after the injection, but we can actually see it for longer times. And when we test the muscles for the protein, in this case, 3 mice out of 5 express dystrophin that is appropriate size for ART101. So this is recombinant dystrophin which the muscles produced, driving by exogenous messenger RNA. When we analyze the muscles by immunohistochemical staining, we see that the protein is not just made in the muscle, but also is placed into the right position, because we see it at the cell membrane, as on the slide up there. Below is the control slide showing that there is almost no dystrophin in the muscle. Here we go for some functional studies because, yes, we now have muscles that express the protein, but does it help to improve the muscle health? And here we see that it does, because when we inject this mRNA into different muscles, actually into 4 muscles, large muscles of the mouse, for 3 weeks. And that works both for 101 and 102, the columns on the right. So the CPK level goes down, and, well, it almost approaches the one of the healthy mice. The level of CPK is pretty high here because the test was done after the exercise, which increases muscle damage. So we do have an mRNA that makes the protein, and that protein is functioning properly, at least in this test. All that was done with local injection. Apparently, if we want to cure the disease completely, we need to find a way to bring the mRNA to all body muscles. So what we do, in addition, is developing a systemic approach. Here we have the result that we see after injecting mRNA in the nanoparticles into the blood. 24 hours after that, we can see that mRNA is in the heart. And we can also see it in leg muscles, gastrocnemius and quadriceps femoris. That was very surprising and very promising because normally, at least as we know, the lipid nanoparticles, they go into the liver and bring their cargo there. They're used quite a lot to deliver therapeutic moieties into the liver. I don't think they've been ever used for muscle delivery, but they work. We just need to see more data about that. We're still finding out how it works on the protein level, which is how much protein is made and how stable is that protein. At the same time, we're doing other things. For example, we're working on those chemical letters that I mentioned before, the letters which make the mRNA. We can use many types of those letters called nucleotides. Nucleotides can bear chemical modifications. So from those shown on this slide, we produced 6 different types of mRNA, which encodes for the same protein. And as you can see above, this is protein produced by in vitro translation. Some chemical modifications increase translation, so we can get significantly more protein from the same level of mRNA. Another thing which we have to consider is the immune response, because mRNA, as any other RNA, is an antigen. When it gets into the cell, it resembles the virus attack. So the cell actually fights foreign mRNA and tries to shut it down or destroy. This is something we want to avoid as much as we can. So, shown on the left, on the bottom panel, is the so-called double-stranded RNA response, because when 2 strands of RNA combine together, they produced a very potent antigen. We can detect this structure by a specific antibody, and we show this on the right. So the same 6 types of RNA we test here for double-stranded RNA. And we see that, for example, B has a high level of double-stranded RNA, as well as A, and the others have much less. On the right, there is a positive control. There is poly-IC RNA, which is normally 100% double-stranded. Here we go. I said a word about those untranslated regions in the ends, and also the cap and the poly-A tail. We can make those elements by different ways. Some of them are easier, others are more difficult. But the thing is that, as in this example here, on the left, there is a line with the RNA that we produce I would say, regularly. So that, when we transfect the cells, gives this amount of protein. But then we made another RNA, again, encoding for the same protein and having the same chemical composition, but it just has a different cap and different poly A tail. It's longer. And we can increase dramatically the amount of protein that the cells make now. So all this brings us to the task that we need to select the appropriate sequence, the appropriate chemical structure, and the appropriate production pipeline to make the RNA that would produce as much dystrophin as possible. A word about the sequences. Here is the structure of human dystrophin. As I said, ART 101 was designed to incorporate as many functional domains as possible. But then we thought that there are forms of dystrophin that are occurring naturally and are almost 100% functional, or even 100% functional, because those people have no symptoms. So we went to the database, the clinical database, and selected 3 types of dystrophin, of this type, of a healthy type. The protein is still truncated. It has deletions inside. And it's much easier for us to make those RNAs than the big one. So we can see here, on this figure, we tested the one which is 6 kilobases and 9 kilobases. So as I said, these forms are natural. They exist in real cells, in real people. We expected them to be more stable, more functional, and produce a significant amount of dystrophin, which was right, at least in this experiment in vitro. And we're working on, as I said before, finding our best sequence among all of them. Of course, as anywhere in messenger RNA therapy, delivery is a very big problem because those bubbles, the nanoparticles, are very complex structure. They're built of many components, and we have to combine them in the proper way to get those ideal nanoparticles. There is one recipe shown there as an example. But actually, when we change the chemistry or we change the sequence, we might need to modify those ratios in order to get the best delivery. It brings us to the need to test about 500 combinations, 500 different ratios. That takes quite a lot of time. Right. By doing that, we can have our ideal nanoparticle. But then again, if we want to achieve systemic delivery, it may not be enough, because nanoparticles would go everywhere. They would distribute in all organs and tissues and probably go to organs like liver or spleen, preferably. So if that happens, we might need to attach other molecules on the top of those particles in order to bring them specifically to the muscle. That could be metabolites which muscles use to produce energy, or there can be special lipids we think about using antibodies or peptides. There's quite a lot of options. But I won't talk about all of them. Just to summarize, we have an external RNA, which is capable to produce dystrophin that is active in the muscle, also in vivo. When we inject that mRNA systemically, we see the distribution among different organs. So there is potential here to bring the mRNA to all muscles in the body. It still remains to be seen how the protein is produced after that delivery method. We have to test different sequences and different chemistry in order to find the best combination. And formulation might need to be optimized for each of those variants in a previous line. So that is the work. At the end, we can come to the point where we could relax, and the mRNA will become a drug and help any DMD patient in the world. Well, thank you. Questions? Well, the question was whether we see toxicity with the treatment that we do in vivo. And the answer is no for now, but we didn't do it for a very long time.